Hi, friends. Thanks for joining us. My name is Marisa Gomez, and I'm the Public Programs Manager at the Santa Cruz Museum of Natural History. And we're really excited to welcome back Christian Schwarz tonight for an evening of mushroom musings and more. And we thought that it would be fun to get our juices flowing a bit and share a quick prompt with you all. So mushrooms are on the menu tonight, and I would love to have you all get your fingers moving, start typing, because we do want this to be a conversation, and share with us in the comments 1 to 3 words to describe what mushrooms mean to you. So 1 to 3 words on mushrooms. Go. I'm excited to see your answers. And for those of you who did try to join us a week or 2 ago, thanks for coming back for more. And a big thanks to our guest of honor, Christian, for rescheduling with us. And on that note, I'm going to welcome Christian in. So one moment. Here we go. Okay. Hey, Christian.
Howdy.
Welcome.
Howdy.
Thanks for being here. How you doing?
I'm doing well. I'm
Great.
happy to be back, and this time with the promise of technological ease.
Absolutely, and I'm very excited to see your full presentation and talk to you some more. Maybe we can get started with you answering our prompt that we've put out to the masses. Mushrooms, 1 to 3 words, go.
Bizarre. I think that has to be first. I think this is my standard response, actually. Bizarre, disgusting, and beautiful. They span the entire spectrum.
I love that. That's
Yeah.
wonderful. And we'll
if they weren't, I wouldn't be interested in them, I'm sure.
Yeah. Yeah, I was thinking about mine. Mine, the first thing that came to mind was odd little surprises.
Odd little surprises.
Odd little surprise. I'm always just
It's a pretty good album.
Surprised by them whenever I see. We've also got people joining in saying otherworldly fruiting bodies and awesome yet gross.
Yes, someone who understands me. Yeah.
Yeah. I think that this person, I know this person, and I think that she particularly does not like to eat them.
Hey, I'm partially there with them. Eating them is not my favorite way of engaging with them.
That's
I think
really
they're
interesting. Yeah,
much
maybe
better
we'll
to
get
look
into
at
that.
and think about than they are to eat.
I'd love to talk with you more as we go on tonight about like the different entry points for people being interested in mushrooms.
Yeah, let's do it.
And before we jump into your presentation, can you share with us just a little bit about who you are?
My name is Christian Schwarz. I'm from San Diego and SoCal originally, which is not a good place for mushroom hunting most years. This year they had a really good season, but I got interested in mushrooms down there and decided that if I was going to go to college, it would have to be somewhere that was in the forest, and everyone told me Santa Cruz was the spot. So I applied and I came here and studied mushrooms so much that I couldn't do school anymore and dropped out and wrote a book with Noah Siegel called Mushrooms of the Redwood Coast, which we finished in 2016. And then I went back to school, finally finished. It took me 11 years, it's fine. And now I teach a class there called Natural History of Fungi through the Norris Center for Natural History, which is the sort of hub for all naturalist interested adjacent students. And I'm a research associate there, so I also do work just on the taxonomy of mushrooms and biodiversity of mushrooms and ecology of mushrooms locally.
Well, thanks for sharing your roundabout way of getting to where you are. I think that it's always good for us to hear that there's not one path. And I also have here your book.
Hey, that's the one.
Yeah, so this is the one, and it is a tome, and it is a beauty. So maybe we'll talk more about this later. But okay, so thanks for everyone who's in here talking with us. And also we've got a new answer to our prompt. Our prompt for everyone who's maybe didn't hear it the first time is, what do mushrooms mean to you? What are 1 to 3 words for describing mushrooms? Write them in the comments. Christian's was bizarre.
Disgusting and beautiful.
Disgusting and beautiful. So we've also got delicious sometimes. Beautiful ecological puzzles. So this is great. Ooh. Please give us more answers to that prompt. And
I like puzzles. That's good.
Yeah. So we are going to get started. Christian has a whole slideshow presentation for us, but also is enthusiastic about being interrupted. And so if at any point during this presentation you have questions or comments or things you want to talk about, put them in the comments. I'm going to be looking, and I am more than happy to barge in and interrupt Christian. And then at the end, we'll have more time for questions. So without further ado, I'm going to let you take it away.
Okie doke. I'm going to get started now. So my face is going to disappear, but my voice will linger on, and I'm going to switch over to my slides here. What I'm talking about tonight, many of you who have interacted with me online know that I am constantly harping about citizen science, and especially the use of iNaturalist to document not only mushrooms, but biodiversity of all kinds, not only here in Santa Cruz, but throughout California and throughout the world, really. But after doing citizen science in whatever way for years, I began to have additional feelings about what we were all doing together. And I started thinking about this term community science that some people had proposed for a replacement for the term citizen science, and what the differences between the terms might be. So tonight I'm going to be talking about that swarm of ideas in a talk that I've been calling Data is Not the Destination. All right, I'll see you later, and interrupt me by sending messages to Marisa. All right, here we go. Let's see. Okie doke. Can y'all see my screen? Yes? Okay, good.
My answer is yes.
Okay. All right. Let's just get started with a recap on what fungi are and why I'm so interested in them. Most of us sort of have experienced this kind of mushroom with a lot of regularity. You see these all over the woods. These are decomposer mushrooms. They break down large diameter logs and the trunks of trees. Sometimes they're even pathogens, rotting the roots of trees. But this group, these conch mushrooms, are almost all woody recyclers, the ones that are common here in Santa Cruz anyways. And they break down dead organic matter, in this case wood, and recycle that carbon and all the nutrients that are locked up in that biomass back into the ecosystem and allow other organisms to use it to fuel their metabolism. But not all fungi are decomposers like this, even though it's what most of us learn in high school or even in college. The main takeaway is that mushrooms are decomposers, fungi are saprophytes or saprobes, but there's a whole other range of things that fungi do in the world. This is really just the tip of the iceberg ecologically. They also can be parasites, like these cordyceps that you see on the screen now. These are growing out of sort of an unidentified mass as far as you can tell from the photo, but what's actually behind them is the cocoon of the pupa of a moth larva. So they probably killed the larva while it was in its cocoon stage, and these are parasites, and there's all sorts of fungi, many of them microscopic, some of them large like this, or relatively large, that actively harm another living organism, whether it's a tree or an insect, or sometimes even people. Like ringworm is not actually a worm, it's a fungus that infects your skin. So there are fungi that are parasitic like this, and they have really important influences on ecosystems by sort of controlling how dense any group of organisms can be. So if there are too many seedling plants in an area, parasitic fungi might thin them out by killing some small percentage of them and allowing them to be more open in the forest. Same with insect populations and so on. And then there's my favorite group, one of the groups that I think is most interesting to research and sort of delve into the literature about the ecology of how they make their living. These are the mycorrhizal fungi. You've probably heard of these. They're getting quite a bit of fame. There was a Radiolab article about them recently, or episode rather. They're even used commercially as soil amendments, and these are the fungi that wrap their tissues, their cobwebby mycelium, which you see at the bottom of the screen, that big white spreading network of fibers. That's really the fungal body itself. That's the part that lasts from week to week and month to month and year to year. And some mushrooms, not all of them, wrap that network of fibers around the root tips of woody plants like shrubs and trees, and they end up forming these little knobby sort of finger-like projections on the root tips that you can see over on the right side of the screen. And those are called mycorrhizal root tips. Myco is the Latin root for fungi, and rhizo is the Latin root for root. So these are fungus roots. And what happens at the site where the mycelium and the root interact is that the fungus takes water and nutrients from the soil and exchanges it with the tree or the shrub for photosynthate, which is just a long fancy way of saying sugar. So the magic trick that plants can do that fungi can't is that they can turn sunlight and water and carbon dioxide into organic sugars, into carbohydrate. And fungi can't do that. They're heterotrophs like you and me. They need some sort of external source of carbon to fuel their metabolism and growth. They need some other organism to get food, whether that's as a parasite or as a decomposer, or in this case, as what we call a mutualistic symbiont. So they're in the symbiotic relationship where there's a mutual benefit for each partner in the relationship, both the tree and the fungus. And the reason that the funguses, the mycelium of the fungus is so much more efficient than the tree's roots at getting water and nutrients from the soil is just a matter of surface area. The finely branched, super cobwebby structure of a mycelium is way more efficient at exuding enzymes in the environment and absorbing nutrients and water than any tree's root system could ever hope to be. So I like fungi not only because they're bizarre and beautiful and gross, like I said at the beginning, but also because they span all of these different ecologies. They're both parasites and decomposers and mutualists. And that's not something you see in every group of organisms. A lot of group of organisms are a little bit less diverse in their ecological roles that they play, but fungi are super diverse in all of the roles that they fill in ecosystems around the world. And not only are they diverse in ecological roles, they're diverse in the spectrum of sizes that they can be. So what you see on the screen right now is a tiny, tiny little mushroom. This fruiting body, which is just to say the reproductive structure, that's the part that you and I see, is smaller than your pinky by a long shot. And this is a little inky cap that was growing out of a single pellet of elk dung. And that was its whole role in the world, was to digest that one pellet of elk dung and release its nutrients back into the environment and reproduce a little bit on the way. And that's what it's doing right now. But even that is not the smallest fungus out there. Baker's yeast, brewer's yeast, the thing that's responsible for bread and beer and wine, is a single-celled, fundamentally microscopic organism. This is not something you can see without a microscope. And these single cells reproduce by budding asexually. So fungi can be as small as a single cell that you need to see using a microscope. And these are also really short-lived organisms. Their lifespan might be something on the order of hours to days. So this is the small end of the spectrum, but like I said, fungi span the entire range of size, scales of size. And this is an example of the other end. These are honey mushrooms, and while they look like normal-sized mushrooms, and they are, it's the underground part of this species that is so astonishing. These reproductive structures that you see on the screen are just the tip of the iceberg. What's going on below ground is a mycelium that might extend, you know, acres and acres, hundreds or thousands of acres across a landscape, and maybe something like 1,000 or 2,000 years old in some of the more exceptional cases. So fungi can be single-celled microscopic organisms that only live for a couple hours or a day. And they can also be these mega organisms that are among the largest and oldest living organisms on Earth, with lifespans measured in millennia, covering entire landscapes, a single contiguous genetic individual as big as a whole hillside or half a mountain range. So this is another thing that really draws me towards fungi, is just how much they span the spectrum of time and space. And they're also everywhere. They are up in the stratosphere in the form of spores, little tiny microscopic reproductive cells that get caught in wind currents and are lifted up tens of thousands of feet above the surface of the Earth. But they're also down in the deep ocean. At the bottom of the screen, there's a hydrothermal vent there, which is a place where water that has sunk through the Earth's crust, through the basaltic layer in the ocean, down deep into the mantle or closer to the mantle, gets warmed up, infused with minerals, and then bubbles back up and steams into the bottom of the ocean. And these are places where you might not ever expect that fungi would live. But if you take a submersible from the Monterey Bay Aquarium Research Institute and go all the way down there, thousands of feet deep, and take a little cupful of that water and bring it back to the lab and streak out some drops of that water on a petri dish, lo and behold, there are fungi that grow out of those samples. What are they doing down there? We don't know, or at least I don't know. What are their names? How do we recognize them? What's the bigger picture of their role in biodiversity? I certainly don't know, but it's amazing to think that not only are fungi way up in the air, way down at the bottom of the ocean, and absolutely all over the surface of the land, fungi are ecologically diverse in their spatial and temporal scales, and just the amount of the Earth biosphere that they occupy. Up at the top of the screen, there's some scanning electron micrograph photos of a fungus called Xeromyces with an X. And that is a fungus that we think has the lowest requirement for water of perhaps any living organism. This is a fungus that lives in the salt crust of things like salted cod that you might get at a Portuguese or Italian grocery store. So this is sort of a way of challenging our notion that fungi are always things that inhabit dark, damp, wet places. Some of them, like this fungus, grow in places that are extremely dry, where any other organism would have a very tough time living. But fungi also live in us, in our bodies. So not only are they all over the ecosystems around us, they are literally in us and on us. They're on our skins, candida, they're in our guts. Some of the microbes that contribute to our gut health are fungi, and they're even in our blood and our brains. There's some evidence to suggest that fungi may have a role in Alzheimer's, and whether it's causal or opportunistic, we don't know yet. But some of the recent research shows that the brains of Alzheimer's patients have greater amounts of fungal DNA than in healthy patients. But despite all of this that makes it sound like we know a lot about mushrooms or a lot about fungi, there's vastly more, and I mean vastly more, that we don't know. There are many basic questions even that we don't have good answers to with regards to fungal biology, fungal diversity, fungal taxonomy. And what I mean by taxonomy is just how to recognize and identify them. So we don't know how much diversity there is globally. Estimates for how many fungi there are on planet Earth vary pretty wildly, from 900,000 on the low end to up to 5 million on the high end. So that's a pretty huge range. But no matter how many million there are, we've only described about 150,000 of them. So the lion's share, like a huge majority of them, remain formally unknown. Their roles in the ecosystem remain mysterious to us, as does their identity. How to recognize them is what that last line is supposed to read. So even if we were to find them all, which we're still having trouble catching up to, learning how to recognize them and name them is an entirely separate question. And then what I mean by distributions, that last point there, is where do they live? Even for the organisms, the fungal organisms that we're most familiar with, like Amanita muscaria, the fly agaric, which some of you undoubtedly recognize, that's the red mushroom with white dots all over the cap, that's on, the subject of many pieces of art in various contexts worldwide. King boletes, a lot of you probably like eating those. Chanterelles, those are really popular. Even those really common familiar organisms, we barely have a good understanding of the map of where they live on the surface of the Earth. So that's something I spend a lot of time thinking about, is how do we build a map to know where these organisms live? Because it controls a lot of the other organisms they interact with, what organisms they'll naturally encounter in the ecosystems they inhabit. And then another thing that is really interesting, especially in the context of the Anthropocene time that we're living in, the climate change context that we experience, is seasonality of mushrooms. For example, king bolete mushrooms that are super popular, edible, delicious, big, beautiful, satisfying to find. Pretty much everyone who lives on the California coast and has spent some time outside has heard tell of king porcini. These are mushrooms that used to be reliably found in October, November. They were sort of classically thought of as a Thanksgiving time mushroom, but they were just found fruiting in Santa Cruz 2 or 3 days ago, in May. We had 0 records of king boletes in May until this year in Santa Cruz. And 2 years before, they were fruiting even in Humboldt County, which is way far north, into January, which is kind of unprecedented. They seem to have this flexibility where they move earlier or later in the year, depending on how warm it is or whether or not the fall was really dry. And a lot of California is experiencing dry falls and sort of wetter springs, a more compressed and sort of lopsided season, We don't know if that's the pattern that's going to continue into the future, but it's something to think about, and it's something I wonder a lot about. How is it going to affect how mushrooms can reproduce in response to things like rainfall and temperature, and whether or not it puts them at risk of extinction? Some mushrooms will do well in the regime of a new climate, and some mushrooms won't. And figuring out which ones those might be ahead of time could give us a little bit of leeway in trying to conserve them or preserve that biodiversity. So these are all things, all examples of things we don't know about fungal biodiversity. But in an attempt to learn more about these things, get answers to some of these questions, to know some of these things, I have sort of focused on using tools like, one more thing, I sort of already mentioned population trends, but that's basically whether a mushroom is at risk of extinction. To get to contribute some small amount of this knowledge that I want to see in the world, I've decided to focus on Santa Cruz County, my home county, small enough that I can drive around it in a day if I feel like it, and really just try and learn everything there is to know about the macro fungi, the mushrooms of this county that I call home. It's a really diverse county for mushrooms, so it's a fun place to do this. There's a long history of people studying mushrooms here, so I've got some work that I can lean on to get me going. And there's a lot of people who love mushrooms here that I can build a community out of. But even in this tiny county, even just focusing on mushroom forming fungi, which means that I'm not paying attention to any microscopic fungi, which is where most of the diversity is, I still don't really have a chance of doing it unless I've got people helping me. And the way I have really centered on is using iNaturalist, which is a tool that comes in two formats, basically. There's a web site that is basically a database with a public face that you can go explore data and see what else has been posted by other people. And there's a phone app that allows you to upload biodiversity observations of any kind of organism anywhere in the world. And the phone app allows you to take a picture and add it to the database that is then represented on the website really easily. In just a few seconds, with a few pushes of a few buttons, almost anyone can make a biodiversity record. And what I mean by biodiversity record is just a record of an organism in a place on a day. So time, place, thing. And just getting those three pieces of information together sounds boring at first, and it is. If it's a single record, it doesn't mean much. But when you get tens of thousands of people, or hundreds of thousands of people, or indeed millions of people around the world making these kind of biodiversity records every day, you start to build a more robust, deeper, more flexible, real-time picture of the world than we've ever had. People talking to one another about what they're seeing is something that I think of sort of as a civilizational sense organ, a way for natural history observations not to be locked up in any one person's head, but for us to talk to one another as a species, as the human race, about what it is that we're seeing in the world that surrounds us, specifically with regard to living organisms. The ecosystems that sustain us are made of living organisms, after all. So it's a really important tool, in my opinion, to be able to talk to each other about what we're seeing in real time. So this is what some iNaturalist data might look like if you were to explore it through the website. This is just Santa Cruz County in that orange outline, and all of my observations of living things in Santa Cruz County. And you can tell approximately where I went to school, that dense cluster of dots, and you can tell the places that I almost never go because I don't have access to them, or I just don't live near there, or find it inconvenient to drive out there. And those are the limitations of any one person's experience of biodiversity in a place. But when I add up everyone's records of biodiversity for Santa Cruz County, it starts to look much different. So I set up this little project to keep track of mushrooms in Santa Cruz County on iNaturalist, and it's really gotten an amazing amount of participation from people I've mostly never met. You can see the statistics on this screen. There's about 1,800 people who have contributed some biodiversity records to our project, almost 900 people who've then gone and helped identify those observations, and they've added 30,000 observations representing more than 1,000 species in our county, and this is just in the 3 or 4 years since I set this project up. So I didn't have to tell anyone to do anything. They just downloaded the app and went out in the woods on their merry way and took pictures of whatever they felt like. And when you scale that up to 1,800 people, you start to get a really rich picture of the mushrooms that live in our county with very little input cost of effort on developing a protocol or telling anyone what to do. It's just fun. People just go out and do it because they feel like doing it. And when you scale this up to the state level, so we went from an individual to a county, and now we're going to the entire state, it's really jaw-dropping. 17,000 people have submitted 250,000 observations, roughly speaking, of about 2,500 species. That is incredible. And this is just in less than a decade, far less than a decade, about 5 years of serious data collection, with no one having any sort of rules about what to do. This is just people going out and observing mushrooms because they feel like doing it. And this really warms my heart. This is the utopia that I want to see. I want there to be this vast network of people connected in tiny little interactions as well as deeper interactions, whatever level suits you, and together building this picture of the living organisms that surround us. And this just represents how much iNaturalist usage has gone up since people started using it for mushrooms, which was in about 2015 was the first year with serious engagement. So we're less than 5 years in, or about 5 years in now, and we've already got this amazing data set. I can't wait to see what it looks like 10 or 15 years down the road. We're going to know so much more about the fungi of California and of hopefully the United States and the world, the more people get involved. And before you go thinking that iNaturalist is just a United States phenomenon or a California phenomenon, that's absolutely not the case. There are really robust growing iNaturalist communities in places like New Zealand, India, Mexico. These are places where there's been rapid growth and sort of the explosion of iNaturalist culture. People are really excited about using it there, and it's sort of even created little pockets of biodiversity enthusiasm and ecosystem protectors in places where there maybe wasn't a lot of value for open land and wild spaces prior to people realizing that biodiversity was sort of all around them in the places they called home. So these graphs just show that iNaturalist is very much in its growth, big growth phase right now. There's really no sign of slowing down yet. And I just want to tell this little story because I find it so fun. This is sort of the best of iNaturalist in my opinion. My friend Ryan, who is username pterodactyl there, you can see on the upper right, is an ornithologist. He studies birds, and he was up in Alaska last summer for a bird conference and had a little extra time, so he went hiking. And there's mushrooms in the summer in Alaska. That's their mushroom season. So he was spamming our friend group WhatsApp with lots of pictures of mushrooms that he was finding, and he doesn't necessarily know what they all are. So he was just sending them to us and saying, hey, look, isn't this cool? And I sort of tuned it out because he was sending so many pictures of so many mushrooms. And when I checked the next day, I saw that he had observed this mushroom that you see on the screen right now, just this one picture. And my jaw dropped because I instantly recognized it as a member of this genus Squamanita that you can see up there at the top of the screen. And I didn't know which one it was, but I knew that it was a super rare, super weird mushroom that I've never seen. Most mushroom experts that I know have never seen it. It's an extraordinarily rare find. And so I said, Ryan, you got to go back. And he said, it's pretty far down the trail. I don't know if I can go back. And I said, no, you don't understand. This is the best mushroom you'll ever find. You're done. Your career is over. This is it. You've peaked. But I need you to go get it. And much to my surprise, he did. He went hiking back down the trail, found the exact same spot, took some more pictures, collected a few of the mushrooms for me, brought them back to his rental car, dried them on the dashboard, and then came back through the airport, got through TSA just fine, sent them to me through the mail from LA to Santa Cruz. I received them a couple weeks later, and I did some microscope work, and I took a little DNA sample, and we got a DNA sequence back within a month that revealed that it was this species Squamanita fimbriata, which had only been seen twice before. This was the third record basically ever of this mushroom, and it was the first report of this species for the entire Western Hemisphere. So this is an example of someone who doesn't necessarily know what they're looking at, taking a picture, being able to connect in real time with someone else's brain halfway around the world, or in this case, just a few hundred miles away, but somewhere far removed from you, getting it in front of the eyes of the right person who recognizes it for what it is, for its significance, and then being able to sort of close the loop of data and get all the pieces moving together in the matter of a few months. You know, we had as much information about this mushroom as we wanted within a month of it being found, or a couple months of it being found. So that was sort of one of the most satisfying things that's ever happened for me on iNaturalist. And it happens for other people all the time in different groups of organisms every year. If you start using it enough, you'll undoubtedly get comments or research requests from people who study particular groups of organisms who want to know more about what it is that you've seen. So what does this show? What can amateurs do? And what I mean by amateurs is the body of the people who spend most of their time on iNaturalist. They're not professionals. They're not researchers. They're not academics in any way. Most of the people there are amateurs, and that's in the original sense of the word, which comes from the French root, I believe, for love, to love. People who just love observing biodiversity. And what has iNaturalist shown? It's shown that amateurs can observe, record, and share with each other biodiversity records in real time in a long format way. People do this year after year over the entire globe. We've got observations from every country. Every continent is represented in iNaturalist, and people do it every day. There's really no gaps in usage. People like doing this all the time as part of their daily practice of natural history, or just as an incidental thing they might be doing while they're taking a hike in the woods or going backpacking. But what can academics do? Say you are some kind of researcher, or you have some position at the university, or you have access to specialized equipment and you know how to use it and you know how to interpret the data that comes out of it. I think we're just at the beginning of knowing what we can do with this big mass of quantified biodiversity data. We don't really know yet all the kinds of questions that we're going to be able to ask from this data that we've all accidentally generated. And this is the case for many innovations in science and technology, is that an innovation comes first, and then what you can do with it comes second. You don't necessarily develop a tool because you had a use for it. Sometimes it creates its own new ways of seeing and its own new ways of asking questions and its own new narratives and its own new communities. So I encourage academics to be really creative about what it is they think they can do with this data. And there's probably no better way to get familiar with it than to become participants themselves. Some of the creators of iNaturalist and I have discussed this before, and they've always told me that the people who are most able to leverage the data that comes out of iNaturalist are those academics who have become users themselves, and they understand the limitations of the data, and they understand the potential and the sort of world of things that can be done with it, and they understand how to interact with the community of people who submit the data. So a lot of you have probably driven up Empire Grade on the west side of campus and seen the Mima Meadow, which is right across from the west entrance. And this is what it looks like on iNaturalist. A lot of people walk that central trail. Some people sort of range out over the mounds themselves, and they've seen, you know, about 400 species, 1,400 observations. But this is really not what gets me up in the morning. This is the data that comes from people using iNaturalist and visiting this well-loved natural space, this open area. But it's not how I want to see this open area. It's not what I think of when I dream about it in the spring or in the fall. What gets me out of bed is thinking about it like this: a beautiful space, a space that sort of lifts my spirits or sort of makes my blood boil with anticipation. It gets me, it makes me throw the covers off and put my shoes on and get up there to see what's blooming or what birds are migrating over. And that's because the summary of all of the organisms I've ever encountered there, the sort of distilled essence, the derivative of my experience, is more like this. It's some sort of hallucinatory aesthetic image of what it is that I feel when I think about the Mima Meadow, or when I think about Marshall Field, or when I think about the Sierra Nevada, or any particular spot that I've got an attachment to. It's a swarm of experiences, both emotional and aesthetic, and to some degree related to the data that I collect there and the research that I might do with what I've seen. But this is the side of citizen science or community science that I want to talk about now. And I mentioned at the beginning that there's this sort of divide between people who sort of got tired of using citizen science as a term and wanted to use community science, and those who still use community science or citizen science. And at first, I wasn't sure that it mattered much. And then in the past 4 years, it's become clearer and clearer that using the term citizen harkens back to a way that we divide ourselves as people that I don't think is particularly good in any context, but it's certainly not what we need when we're talking about a civilization sensing the world of living things around it. I don't care if you're a citizen of anywhere, and I certainly don't care if you're a citizen of the same political entity that I am. What I more care about is the community of people who love living organisms, regardless of where on the planet they live. So I've started using community science, not only for that reason, because I feel like it's much more inclusive, but also because the community in some ways is what I really need in the end. I don't necessarily need data. I don't necessarily need to do research with what people are going out and seeing. I would be actually quite happy just to see what people are seeing, just to share in their observations, just to have access to their mind, to be with them virtually in a way that I was never able to before, to see a more visceral sense of what they're experiencing in the world. And the reason that I want this is a number of sort of axes of why this is important to me. If we lower the barrier to entry and allow people to participate in community science by doing whatever it is that floats their boat, whether they're watercolor artists and they just want to document a few species here and there, or someone who wants to download the app, make one observation, delete it immediately and never use it again, or if you're a power user, someone who goes out and documents 20 species a day or makes 10,000 observations a year, all of those people should have some place, some home in this community of people who spend time observing the lives of non-human organisms. And that's really what I mean when I say a naturalist. Someone asked me this recently, what do you mean when you say naturalist? And I realized it was, in my head, really just someone who spends a significant amount of their time being attuned to non-human organisms, finding out what their lives are like and who they are and what they do. And iNaturalist has really managed to lower the barrier to entry to participate in this community. A lot of the previous or prior citizen science platforms weren't particularly easy to use, and it weeded people out. People didn't feel like using it. iNaturalist is so easy to use that it's gotten the benefit of all of these different brains, all of these different people with their particular aesthetic attractions, with their particular desires for what they go pay attention to. And that makes for a much richer picture of the world. And when the barrier to entry is low like that, it allows people to play. There are some people who really just have fun with it. They go out and document every single saguaro cactus they find and no other organism. That's what they find satisfying. It's what they do. And so now we have, at least in that one part of the world, an exceptionally detailed map of where saguaro cactuses live. It's a very compelling organism. You could see why someone might get obsessed with them, but that's the kind of thing you get when you have lots and lots of people participating. There's this neurodiversity that humans always bring to the table. And the more minds you involve, the more likely you are to get some people doing weird and wonderful things with their attention. And what this ends up looking like is knowledge creation by accident. There's no rule book, there's no binder of instructions, there's no end goal. We are accidentally generating knowledge together by playing in a space where there is a low barrier to entry. And that brings me to the title of the talk. There is no destination, and it's certainly not data. There is no endpoint to nature. There is no endpoint, at least hopefully in the near future, to our human attention that we can leverage, that we can direct towards the outside world and to living organisms. We're going to do this for fun forever, at least in my utopic vision. That's what I want to see. And I spend a lot of time teaching people, both in classrooms and out in the field, and this is what I see. People who are making iNaturalist observations, but I don't think they're doing it by and large because they care about where the data goes, or I don't even think they have a sense of what that particular data point will ever be used for. In fact, I don't think that they're under the impression their individual observation, any particular data point, will ever be used. I think what they really like is seeing these organisms, being delighted in the aesthetic beauty of these critters. This is a picture of one of my students in Big Basin, taking a picture of a little nest of mushrooms of 2 different species that are dark blue and bright orange, and they just made this incredible scene glowing in the dark redwood duff with green moss all around them. And I think this is the kind of scene that really brings people back not only to the woods, but to the community of people submitting what we call data, but are really just little artifacts of attention, little artifacts of appreciation, little artifacts of beauty that people try and document. And this is what it looks like when I'm out in the woods with people. It's people with big smiles on their faces, or their heads tilted up in awe at a giant California nutmeg tree, or cradling a little mushroom in their hands and marveling at its structures that they'd never seen before. And that's what I really think biodiversity appreciation is, what naturalists really feel when they go in the woods. This is sort of what distinguishes it from science. Science might go out with a question in mind and collect data in a way that might be quite arduous, really not how you want to direct your attention, and you wouldn't otherwise do it if you didn't have a question you really wanted to answer. Whereas what I call natural history is much less directed, and it sort of leaves you open just to turn your aesthetic desires, your attention, to whatever part of the world calls to you. Just another view of people poking, prodding, and appreciating mushrooms out in the woods. So what I think brings people to the community science community is aesthetic delight, fascination, and wonder. And I think these are things that intuitively sound right. This is what I think a lot of you probably acknowledge brings you to the woods over and over and over again. And just as an example, and as a little plea here, this is up in the high desert of South America. There's these giant salt flats, dried out old lake beds with big crusts of white salt in between low vegetation in this extremely harsh environment at thousands of feet of elevation, where there's mountains and snow up above. And that arrow that you see on the screen is pointing at a vein, a seam of bright orange mushrooms that were recently discovered there in the past handful of years. And here's a closer view of them. These brilliant orange mushrooms erupting through bright white salt crust at the edges of these lakes that are inhabited by flamingos. Just in case I don't get to write a will before I die, I want someone to sprinkle my ashes in this kind of terrain so that my nutrients and my molecules will be integrated into a flamingo's intestine somewhere. This is the kind of wildly beautiful landscape that is super bizarre. I can't even believe there's mushrooms there, but this is where I want to be directing my attention in the world. This is the kind of thing that I want to surround my life with. And there's other people. Here's my friend Allison, who takes macroscopic photos, macro photos of little slime molds, and the tiniest mushrooms you can imagine are her quarry. That's what she really likes doing. And so in the past few years, her articles have been featured on This Is Colossal, all sorts of places. I think she even had a few photos featured in National Geographic. And she is not someone who's primarily interested in identifying slime molds. She doesn't necessarily care. I mean, she likes to know what they are, but her real goal is to bring them to life, to bring them to a size that you would never see them out in the world with your 5 senses. She likes showing you exactly what it looks like if you were to be as small as a springtail or a little isopod. And so thanks to her, we've got this amazing library of photos of slime molds that we otherwise might never have seen. And so this is what I mean when I say that attention is not likely to end soon. We are a part of this big chain of humans, people with consciousness, people with desires and attention that we can direct outwards at the world and share with each other what it is that you've seen, what it is that you find compelling, what it is that you find beautiful. If you are a naturalist, actually, even if you're not a naturalist, as a human being, your gift is your attention. And as naturalists, our gift, that gift of attention is something that we can bring to one another and say, hey, look at this cool organism here. And that's what I want more of. We receive artifacts from past naturalists who've told us about the beauty that's around us that we can go looking for, and we can leave artifacts for the future. I really am heartened by all the people who do nature journals, who do nature illustration, who write about their experience in the outside world. And my form is to leave a detailed legacy of iNaturalist observations, all of the organisms that I saw around me. Thankfully, we have a poet laureate of community science, Mary Oliver, who knew all of this way before I did, and she wrote a little poem about it that ends like this. Instructions for living a life. Pay attention, be astonished, tell about it. 7 words at the end there that give you all the instructions you need to be a community scientist, someone who goes out and pays attention to the natural world and shares it. And how could you fail to be astonished? I think Mary Oliver knew this perfectly well. If you go out in the woods here in the spring, you might see these lime green and salmon pink waxy caps growing right next to each other. Absolutely gorgeous. Something you might never believe you'd see out there until you saw it right in front of you at your feet, defying you to deny its existence. Look at that beauty. Moths like this, it's hard to believe that they really exist. You can't help but be astonished. The world is flabbergastingly full of organisms that make your jaw drop. Nudibranchs, this is an organism you might see right here off the coast of Santa Cruz in a tide pool. That red color of the antennae, so to speak, at the top of the head, that purple color of the body, the orange color of the gills, that's all one pigment bound up with different metals or in different formats that actually refract light differently. These are the kinds of mysteries, the kinds of puzzles that we can start by being attracted to because of the beauty, and then do a little bit of science, go a little deeper, do a little bit of extra research and learn these sort of puzzles or mysteries about the natural world around us. A bird like this, blue and pink and yellow and white and black, looks like a beautiful little clown. You can't help but be in love with nature once you start paying attention to it. You can't help but say, that beauty is sublime, that beauty is transcendent. Look at this fish. It defies description. I love this fish. But loving these kinds of organisms, being astonished by them, finding richness in them, and incorporating that into your life is a risky thing to do in this time in the world. You are signing up for front row seats at a kind of bonfire where priceless works of art are being thrown in every day. And this can be a bit lonely. Not everyone out there understands just how deeply beautiful the natural world is, doesn't necessarily feel the same way that we do about it as naturalists. And we face mass extinction, defaunation, and homogenization. And those last 2 words might not be familiar to you, but defaunation is just, even if an organism doesn't go extinct, there might be a lot less of those organisms. So something like a pronghorn antelope, they're not extinct, but there's nowhere near as many of them now as there were 100 years ago. And homogenization is this idea that invasive species, humans transporting organisms around the world, starts to make it so that these richly flavored, sort of distinctive natural communities that we might have grown up with start to look more and more homogeneous, more and more similar to one another because of invasive species and because of extinctions. So this can be pretty traumatic for someone who is a naturalist who really invests in paying attention to the lives of non-human organisms. Just a few weeks ago, well, I guess months ago now, scientists at the Ocean Sciences meeting in San Diego predicted there might be a 70 to 90% decline in the extent of coral reefs in the world in the next 20 years. And that is shocking, terrifying, stunning. That's going to be something that's going to be really hard for a lot of people to cope with. The number of birds in the United States since 1970 has declined by about 3 billion. That's a massive decline in the number of birds that we think are occupying the air above us, singing in the bushes around us. And it doesn't end there. There's probably this, you've seen this report on insect abundance, not necessarily diversity. They may not even have gone extinct per se, but there's just fewer insects than there were a few decades ago by a lot. And when you start experiencing declines in the number and diversity of organisms down at the bottom of the food level or the food chain, these sort of lower trophic organisms like invertebrates, that's bad news for the functioning of the ecosystem at large, for the habitats that support us as humans. Even fungi are not immune to this. There's a predicted decline of about 30% in the diversity of fungi in the temperate forests of North America. Came out recently, just this January. So what I want to find is a way to turn this sort of thunderstorm of emotions into something that, as a community, we can deal with. How do we sort of find each other, talk to one another, appreciate nature together, but also find solace together? How do we help each other deal with the loss of biodiversity, with the loss of richness, with the loss of aesthetic delight? Biodiversity has always been a nucleus for human communities, but it needs to become that more and more. That is something I want to invest in. I want to see naturalists continue to close this loop that involves science and culture on sort of polar ends of the circle, and just make science feed into natural history, feed into art, feed back into science, and make it so that biodiversity, wild organisms are a part of our aesthetics, are a part of our celebrations, are a part of the stories we tell each other. I want wild organisms in all their diversity, not just the big showy ones, to be something that we celebrate as communities of people around the world. And even on those down days, when you've heard about some extinction event or some ecosystem being wrecked, beauty will still be there in the natural world, and it will still be a balm. You can still sort of immerse your brain, immerse your attention in those patterns that seem unbelievable and those colors that are incredibly vibrant. Even in your backyard, you can find the intricacies of an insect crawling around on your roses and let your brain go into that for a while. Let your attention dissolve into the beauty that surrounds us. And it works, it helps, it makes me feel better. There's always gratitude to be had. And there's perspective that we can use to help us as well. These things are all fleeting. The Anthropocene will be a very short blip in the overall trajectory of this world. There's more things coming. Nature bats last. Biodiversity will always gain the upper hand in the end. And it's really ourselves that we have to look out for. Our desires for an intact ecosystem, to prevent biodiversity loss, to prevent defaunation. We're sort of looking out for our own aesthetics. It is not nature who will fall apart in the end. It is ourselves, our health and our hearts. And that's what we need to communicate to one another. There are bright spots too. There are more whales in the oceans now than there were 30 years ago, thanks to bans on whaling over most of the world. And you can now see bajillions of humpbacks, it seems like, at the right time of year in Santa Cruz. Lots of gray whales. Whale populations are doing better. So you can always look for the bright spots. There are gratitude points to be had. Gary Snyder wrote a little poem about how we should go forth in this sort of troublesome world. And once again, in very few words, I think almost the same number, 7 words, same as Mary Oliver, gave us some instructions to take with us. Stay together, that's the community part of it. Learn the flowers, that's appreciating biodiversity, and go light. Minimize our impact, be a little more conscious about how we tread on this world, what sort of footprints we leave. So I try and keep that in mind when I structure my classes at the UC. Natural History of Fungi is a class where we do want to do science. We do want to learn about biodiversity in a sort of sciencey way. We do want to research biodiversity, but we also want to go light, stay together, and learn the flowers. All right, I'm back.
Thanks, Christian.
Yeah.
Wow. So we've had a lot of comments during this time. Many are just things like, look at this fish. And some suggestions for other places to look for information on citizen science and community science. And so I'll get to some of those musings, but I'm curious myself if you could talk a little bit more about this relationship between the aesthetic world and scientific understanding and what that means for you. And how has your work in collecting images and understanding, have you turned that into art in any way? Have you had people turn it into poems? Have has that happened at all within your little world?
Absolutely. There's always events at colleges, at museums nowadays that sort of claim that they're bridging the gap between art and science. And they are, but that has never really been a gap. There's always been people operating in that space. There's always been folks who are a little bit sciencey and a little bit arty. And if you go deep enough into science, you find out that science really is just a kind of highly derived art. There's way more of that than probably academics want to admit, but it's art. It's totally art. There's never really been a meaningful divide. It feels like it for reasons that matter, but I think those reasons are becoming less and less salient in the world that we live in now. There's people who are doing science and doing art in full-on ways. In a perfect, the Venn diagram has become a circle. There's really no areas where they don't overlap. Juniper Harrower is at UCSC. You've probably met her. She is someone doing that. But the examples go on and on. I mean, there's people in Southern California. I know someone who is a botanist who makes just beautiful designs for shirts and fabrics out of native plants. There's people here who are, some of you have probably been down to Studio Abachar. He's a local who paints fish, and they're works of art. They're beautiful, but they're also scientifically useful. They show up in field guides. They show you how to recognize these fish. There are people who include poems at the start of their big, massive tomes on seabirds of the world, or, you know, if you've ever looked at Milton Love's postmodern experience, Fishes of the Pacific Coast, it's pure art and it's heavy science at the same time. There is no difference.
It seems like your way of approaching your own work too, like the entry point isn't necessarily a means to an end. It's like all parts of your life are impacted by these experiences, and sometimes it feels like art, and sometimes it feels like you're contributing to scientific understanding, and it's the journey.
And it's also a feedback loop. If you pay attention to it through the lens of art, it's going to make you pay attention to different things when you're doing science. And when you're doing science, it makes you pay attention through a lens that will then inform your art. There is no inner partitions in our brains, and those things bleed over to one another. We end up looking at the world differently pretty much anytime we think a new thing about the world.
I will ask a specific question now from someone who's watching, which is about the mushroom in Chile. I think it was in the salt flats. Just curious if you have like an ID.
Oh, it's called Lichenomphalia altoandina. Altoandina means high Andes, so it grows in the high Andes.
Do you have any? We were also kind of like chatting about bucket list mushrooms in here.
That's one of them.
Do you have any bucket list mushrooms?
Oh my God, I have a lot. I have a whole file on my computer called Stuff to See. And it's not just mushrooms. It's like plants and animals and a lot of mushrooms. But I definitely have bucket list mushrooms.
Do you feel like you would know it when you see it? Like, have you learned enough about them where you would be able to recognize it? Or is it possible that you would stumble upon something and then upon further reflection be like, oh my gosh, that's
well, a lot of the stuff that's on the bucket list is there because it's so distinctive. Like, it's just so bizarre and weird that it would be hard to mistake for anything else. But then there are things that are outside of my knowledge zone, like certain insects where I'd probably have to do a little research to be like, okay, I definitely saw it. And there's definitely plants like that. There's plants that I really want to see that I wouldn't be able to just ID right off the bat. I'd have to maybe put them under a microscope or draw them. I find that drawing often directs my attention to the feature that I need to confirm what it is.
Do you ever show your drawings to others, or are they specifically for your own understanding?
Usually I do them as a means of keying. So they're written in the margins of my keying notes, but sometimes I show them to other people.
Also, we have a comment about in flabbergasted that it's a lichen, the Chile, the Andes mushroom.
It's not a lichen per se. It's got lichen in the name because one of the mushrooms that it's related to often grows in and among lichens, but I'm not sure that this one in particular is a lichenized one. It might be, but I'd have to check on that.
Okay. Also, I'm a little curious about like you as a naturalist and your entry into the world of mushrooms. Was it that, and also the fact that you are interested in birds and you are interested in plants and all these other and moths and all these
anything alive. Yeah.
And yeah, is it that you like kind of get enamored with one area and then you kind of get distracted by another and you leave it behind, or have you always sort of been scattered in your interests?
No, at first it was actually tide pool organisms. That was my very first interest in well, actually fish, and then intertidal organisms, and then mushrooms. And once I got to mushrooms, it was all mushrooms all the time, like wall-to-wall programming for like 10 years. And then I started like realizing that there are dry seasons and you have to do something when it's not raining. And so I started paying attention to birds because I had a bunch of friends who were birders. And I forget what happened. They're usually like discrete individual moments that like turn me on to a group, like dragonflies or moths or whatever. And then after that, it's just seasonal. Then it's like, okay, what's happening right now? And so in the winter, it's mushrooms. And in the spring, it's plants. And then in the summer, it's dragonflies. And in the fall, it might be tide pools.
Yeah.
So it's very, I don't ever have to try. Like the cycles that are natural direct my attention to whatever is sort of bubbling at that moment. What's good? What's cooking?
Yeah, I can relate to that. That's also our programming. There's certain
Yeah,
things that come around every year. We talk about amphibians in the, late winter.
Right.
Often. And yeah, now we would be going on wildflower walks, but instead we're sharing pictures of wildflowers. Yeah. I have a couple questions that were like from offline.
Okay.
That people wanted me to ask you, including if you could be a mushroom, what mushroom would you be?
There's so many different ways that I could answer that question. Some mushrooms I'd want to be because I know what they do, and some mushrooms I'd want to be so that I could finally get a window into what the hell they're doing. So I might really want to be a cordyceps. Oh wait, do you guys know about the 8 o'clock howl?
Oh yeah.
It's happening right now, so I'm gonna howl real quick.
Okay.
I'll be back with you.
1, 2, 3.
Oh!
You guys all have to, right?
All right, I'm back. It's like one of my favorite things. I really like the 8 o'clock howl.
It's really delightful. Yeah, it's a bright spot. Cordyceps.
Yeah, so cordyceps, because I don't really understand what it's like to take over an ant's brain. I mean, that might not even be that fun, but I would learn a lot, I'm sure.
Yeah, you don't know what that feels like?
Yeah, I don't, surprisingly. I don't know what it's like to take over an ant's brain.
Well, I wonder too, so if you take that angle, I mean, could it be that you just choose like being not the mushroom, but the entire organism so you can get a better understanding of mycelium? Since that's like,
yeah, I would definitely love to understand what it feels like to be a mushroom year to year. Like what happens when it dries out and it's not reproducing? Is it just like scavenging out in the forest floor? Is it growing its mycelium around the root tips? Does it feel hungry at that time? Or exploratory? Like what goes on? I would love to know what it's like to be a mushroom.
Me too. I just remembered one thing I wanted to ask you about your previous statement about how it started with fish.
Yeah.
Just in the natural world. Was that because you liked to go fishing?
I didn't like to. I was asked to choose a hobby for the summer, and for some reason I chose fishing, even though I'd never done it before. I was always kind of intrigued by the idea. I mean, I like them as organisms. They're weird. Like they have scales and slime, and they're definitely like outside of our element. They're like in the water, so they seem kind of foreign, which is attractive. And so I wasn't a very good fisherman. I'm not good at it still. But the few fish that I did catch, I was like, wow, these are cool organisms. I just want to spend my time in tide pools. And just started paying attention to everything else that was around them.
Yeah, I feel like that's a lot of people's entryway into becoming naturalists, is that it starts with some either use or
kind of
like
repeated
exposure, and the motivations change. So it might start with motivation that's not that aesthetic value that you're talking about, not just paying attention and being a part of natural world. It's about like a resource.
Yeah. And,
right?
that's sort of just a different kind of aesthetics, right? It's gastronomic aesthetics.
Yeah, that's true. And,
I did that for a couple months. Like at the beginning of being interested in mushrooms, I really wanted to eat them, but it almost instantly switched away from that.
Yeah, that's it. I'm curious if anyone watching wants to drop a line of why you're interested in mushrooms, if it's because you like to forage or if you use them for some reason, that might be interesting to hear. So please feel free. And also, I'm wondering if you have anything to say, Christian, about everything that's going on right now, pandemic land, and if, that's impacted your perception right now about being a naturalist, about, exploring the world around you.
Yeah.
Or
you may have seen it before, and maybe some people who are watching who've been to the fungus fair in the past couple of years have seen the talk that I give called "The Most Dangerous Fungi in the World." And it really is about what's happening now. I didn't know this was coming, but I had started talking about this idea that disease is a naturally occurring organism. Disease-causing organisms are out there. And the only way that we have a chance of sort of getting ahead of them, keeping up with their emergence and their effects on civilization, is to be doing community science, to be proactively paying attention to what's happening around us and responding to it in real time. That's exactly the sort of thing that sort of screening, monitoring, testing should have looked like. It should have looked like citizen science. It should have looked like people with a low barrier to entry, some sort of portable, rapid, easily disseminatable test, going out and not having criteria at the beginning for who gets a test, but just doing broad scale testing. Right at the beginning of all of this, I heard that they were not allowing certain people to be tested in parts of Washington because they didn't meet the criteria for being high risk for COVID. And as a citizen scientist, I thought that is 100% backwards. You don't set up the criteria at the beginning. That is when you need to do broad spectrum sampling to figure out what it is you're dealing with. Unfortunately, that was limited by the fact that we just did not scale up tests at the right time. And these are all the kinds of things that someone who cares about monitoring things in real time, whether they be organisms at risk of going extinct or changing under climate change regimes, or pathogens, risks to human endeavors, these are sort of obvious. You need to have your diagnostic tools ahead of time as soon as you have noticed that you might need them. So we should have scaled up production of testing a long time ago and then done just complete shotgun scattered testing of anyone with no criteria involved right at the beginning. That's my opinion. I'm not an expert on this, but it felt like, as someone who does citizen science, who's interested in detecting things in real time, you don't set up the sort of narrow gate at the beginning of the phenomenon. You keep it wide at the beginning and narrow it later.
Yeah, which is what we've been talking about, is, you know, and to a certain extent, not really looking for anything in particular when you go out, and then and not knowing what the benefit's going to be of that data, and being surprised by it, like your friend finding that mushroom in Alaska and not.
You didn't know he was going to find that, yeah.
And it could, you know, be an important piece of the puzzle.
Yeah.
So yeah, so even if we don't know, even if we're doing it just because we are curious and because we are intrigued and because we feel attracted to something, we don't need to know the greater benefit that could come from that. And there doesn't have to be a greater benefit.
Right. There doesn't have to be, and it's still available to be used if there ends up being one.
Yeah. Do you have any advice for people who have not yet started using iNaturalist? Any words of encouragement for getting them going, or maybe an alternative?
I don't have an alternative. It really is the best platform that I've used by a long shot. Like, it's not even close. I have one practical piece of advice and one just sort of words of encouragement narrative piece of advice. So the practical piece of advice is that once you download the phone app, turn off your automatic upload. Go to your settings, turn off automatic upload, then it won't try and use your cell data. It'll just use Wi-Fi to upload your pictures. That's practical. My words of encouragement are that you really don't need to know what you're looking at. You also don't need to take pictures of things that are rare or special or uncommon in any way. We really want observations of everything all the time. Common species, over and over again. People will say, oh, I've already documented, you know, with this sunflower or whatever it is. I don't need to submit another observation. But every data point adds up. The relational value is what iNaturalist is all about. Getting documentation of one organism through time, many organisms once or twice, you know, all of that matters. It's all helpful data. If it draws your eye, if it catches your attention, document it. Don't worry about what it is. If you don't know, we'll help you figure it out.
Yeah, and I think for me, I find that it helps me to better get to know the things that I'm documenting if I am able to do it time and time again, and I'll like recognize the name and be like, oh, I saw this last week, and So I've been documenting the same things over and over again in this one, just at the museum's garden while I'm out there weeding. And
It's
I've
an
got to know, I
amazing
had no
personal
idea that
diary.
there was like 8 different species of ladybugs in our front garden. It's like blew my mind. And then I'll see one like, day after day. And then the next day, like 4 days later, it's like all convergent ladybugs.
Wow, different lady beetle.
I love it. And also we had some answers to our why mushrooms. So I'll just share them with you. Some people started with foraging and it turned into a compulsive obsession, which I'm sure
I know that. Yeah.
And then I love mysteries and puzzle solving and collecting. It's basically a scavenger hunt from nature with accompanying puzzles to solve if you're looking, aka the most fun thing ever.
Absolutely.
So that's like words to live by.
I jive with that.
And maybe that's a good place for us to leave it. And we've got more thoughts coming in, so people keep putting down why mushrooms for you. And thanks so much for joining us, everyone. And especially for those who tried to join us a couple of weeks ago.
Thanks for sticking with it.
Christian, do you have any parting thoughts that you want to share?
If any of those questions come in, you can keep sending them for a few minutes. Marisa, you can send them to me and I can answer them on my Biodiversify Facebook page. So maybe I'll just make a little post with whatever questions you guys end up sending in that Marisa sends to me.
Yeah, and so yeah, Christian has a Facebook page, Biodiversify, and also an Instagram account, Biodiversify, and you can follow him on iNaturalist to see what he's seeing in the world around him at Leptonia.
Yep.
Is Leptonia your favorite?
It's one of my favorite groups of mushrooms for sure. Yeah.
Okay. Well, thanks again for joining us, everyone. We will continue to have more live programming from the museum. We have a weekly live program now that's every Wednesday at 3 with 2 geochronologists from UC Santa Cruz. They do every week a different entry point into geology. And we're starting like very, very entry. So no matter where your interests lie with rocks, you can join us every Wednesday at 3 and ask your questions, and they're happy to ID as well, and more to come. So thanks again for spending your evening with us, and goodbye.
Bye, everybody.